Yeah, hi, welcome to this introduction to metabolites. Hi, I'm Thomas. I'm part of the metabolomics team at EMBL EBI. EMBL is the European Molecular Biology Laboratory, and EBI is the European Bioinformatics Institute, and I'm a curator for metabolites. And the aim here really is not just to make you familiar with submission, but also comfortable to consume and reuse studies. You know, every day new data are made public on metabolites, and it's primed for being repurposed or concatenated, and so on. A little background to begin. The goal of EMBL EBI is to ensure that the growing body of information from molecular biology and genome research is conveniently accessible to all facets of the European scientific and biotechnology community in ways which promote scientific progress and global competitiveness. So that is just making biological-based data open source and available to all. So the omics generates a vast quantity of data, and of course metabolomics is no exception. And metabolomics is still a developing field, so what we interpret today may be expanded tomorrow. So learning from the other omics that came before, we tried to stay ahead of the curve. So that is to develop good protocols and reporting systems, to help support the integrity of the data, to help develop tools, and just to make research available for future developments. Recently, you've heard a little bit about the FAIR principles were developed, and this was to address an urgent need to improve the infrastructure, supporting the reuse of scientific data. So a diverse set of stakeholders representing academia, industry, funding agency, and publishers came together to design and jointly endorse a concise and measurable set of data principles, aka the FAIR principles. And this was really a specific emphasis on enhancing the ability of machines to automatically find and use data, in addition to supporting its reuse by individuals or humans. So examples include to make data findable, accessible, interoperable, and reusable. So you would need unique identifiers, unique or persistent identifiers. You'd need index data repositories, data associated with their provenance or traceability. So this is rich, accessible metadata. You need terms searchable by humans and machines, and you'd also have control vocabularies or ontologies. So the question was, can metabolomics learn from the other omic techniques and not repeat many of the same mistakes that have been made in the past, which a lot of data was collected, but little information was gleaned? Well, in 2007, the metabolomics community, led by the Metabolomics Society, came together with the Metabolomics Standards Initiative guidelines. The focus was on community agreed reporting standards, so minimum information checklists and data exchange formats. And really the aim of the MSI was to provide a clear description of the biological system studied, as well as all the components of a metabolomic study, so as well as to allow data to be effectively applied, shared, and reused. So since then, Metabolights has used the MSI guidelines to develop a data management method for metabolite study deposition. And while it's great to develop all these standards and encourage data sharing, there needs to be a platform to make the data accessible to everyone. So Metabolights, found at ebi.ac.uk/metabolites, is open source and comprised of 2 layers. There's a study repository, which enables the metabolomics community to share findings, data, and protocols. And there's a reference library of curated knowledge about metabolites that links to the database CHEBI, the chemical ontologies of biological interest. At Metabolights, our mission is to provide the scientific community with a comprehensive, high quality, freely accessible metabolomic resource. But we also develop training courses and other resources for the community, as well as actively promote metabolomics research in general. So as one of the largest set of publicly available metabolomics data, we see Metabolights sees users from across the globe. So the USA, China, Europe, and across academia, industry, healthcare, governments, nonprofits. For example, the UK NHS, the National Phenome Centre, Metabolome, the Pacific Northwest Laboratory, and so on. We Metabolights also see submissions from across the globe. So again, China, the USA, the USA, the UK, Germany, France. So over 99 countries in total. We're seeing submissions volumes increase annually. So last year there was close to 1,700 submissions. We host data across 6,558 different organisms, and that's 90 terabytes of data in total, with a significant proportion human-based research. And studies hosted in Metabolights support publications in journals such as those from Nature Publishing Group, PLS, Frontiers, PNS, as well as more specialized journals towards metabolomics. And it's the recommended metabolomics repository for a number of leading journals. And we work closely with our counterpart databases in the US, so Metabolomics Workbench, and Japan, Metabobank. At Metabolights, we capture all metabolomics data. So that's across the entire workflow, across species, be that animal, plant, or bacteria samples, across technique and platform, so mass spec or NMR analysis. And we're constantly evolving to support the latest advancements. So this is a continuous open discussion. And as mentioned, our requirements are guided by the community. For example, the standards, the Metabolomic Standards Initiative guidelines. Most importantly, we are an archive. So we collect the raw data together with the sample and protocol details, so that the data can be reused and reanalyzed by anyone. A rich data source you can access without having to go into the lab. The primary requirement for study submission is raw data, supported by rich metadata, describing samples, describing the study, samples, assays, and metabolites. And where possible, the metadata capture is underpinned by controlled vocabularies or ontologies. We use resources such as OLS and bioportals, and these are repositories for biomedical ontologies that aim to provide like a single point of access for the latest ontology versions. And this is important because it really adds value to your studies, with the addition of expert curation, confidence, and evidence attribution. So let's take a closer look at metabolites. There are various ways to find information through the Metabolights website. From the homepage, you can search or browse through studies and metabolites. You can browse areas of interest, including studies, compound, or species. You can view the model organisms to find specific studies and compounds. And you can also search based on specific terms and free text. Looking specifically at the study repository, so as mentioned before, you can search based on specific terms and free text and browse the areas of interest from studies, compounds, and species. And within studies, you can go a little bit further and filter based on technology or organism part. And the results will display key information about the study title, the unique identifier, the size of the study, who the submitter was, the target organism, and any study factors. And you simply click on the title or the identifier to view the complete study. So what does a metabolite study look like? Well, here's an example of NTMSS2022. One of the first things you will see will be the study status. When you first submit your study to Metabolights, the status will be highlighted as submitted. The study will then progress through to in curation, to in review, and will finally become public. Next to the study status, you will see the release date. So this is when the study became public. Underneath, you'll see the study title, the submitters, an abstract, and a box to highlight the species or organism, and the option to link to the associated publication, which can be updated at any time. And what I always hear is that linking data sets to publications is an absolute pain. Following this, you'll see a section of tabs that will guide you through all the study information. So we have sections for descriptors, protocols, samples, assays, metabolites, and files. In the first tab, descriptors, you'll find keywords. So this is describing the study, really important search terms, as well as an overview of study factors. And study factors are variables that help describe samples as part of experimental design. Where possible, these keywords and factors should be linked to ontologies or controlled vocabularies. This is guided by the online submission tool, and options will appear for you to choose. Next, in the second tab, protocols, you'll find all the information related to experimental protocols. The level of detail required in the methods and material is identical to the methods and material section in a paper. So for example, you'll want to say where your samples came from, how they were extracted and prepared, through to how they were analyzed, the treatment of your data, and the methods used for identification. And these protocols are really central for reproducibility, right? In Metabolights, they're linked to the assays with important parameters used as tags to help improve, data fairness. So the readability and reusability of your data. In the third tab, samples, you will find specific text mineable information related to your samples. There is only one sample sheet per study, where each row is a distinct sample. And this should include rows for QC, any control samples, or any standards as well. And the minimum information required about a sample is represented in the name and characteristic fields. So these are columns for the organism and the organism part, as well as sample type. And as before, submitters are encouraged to add additional information in the form of factors, such as disease or gender, time, or treatment. In accordance with the Metabolomics Standards Initiative, templates were also developed for the deposition of more specific information. So this is depending on the sample sources, such as clinical or in vitro or plants. We're implementing these in the submission tool on an ongoing basis according to demand. Again, Metabolights uses ontologies for the samples, assay, and metabolite sheets. So with the new website and online submission tool, this is all hidden from the standard view, but it's obvious if you've downloaded a file to edit offline in Excel or used for the expanded table option. And typically there are 3 columns used to describe this. There are 3 columns are included for each ontology supported field. So the first one will be the term of interest. So this will be your organism, for example, Homo sapiens. The second one will be for the ontology used, so the NCBI taxon. And the third one will be the direct link to that word in the ontology. And why is this important? Because in the past, we asked people to name the species in MTBLS1, and we got 8 different answers. And this is especially diverse when we start talking about naming metabolites. So in the 4th tab, the assay sheet, we'll find, again, specific text mineable information related to assays. So for each sample, the specific instrument information is provided in detail. For example, the chromographic system, the column specifications, the mass spectrometer, and it together it's included with the data files. So this is the raw or the derived data files, and it really just gives the consumers the opportunity to download and reanalyze the data effortlessly without entering the lab. If you take a little closer look at the assay sheet, you'll see it's divided into sections. So this is starting with a protocol ref column, which again describes the specific parts of the assay or protocol, the assay and protocol, with controlled vocabularies. And again, this improves your data fairness. So studies with the same approach are easily findable, accessible, interoperable, and reusable. So in this specific case, it's an LCMS positive mode HILIC assay. And when you add a technique, there will be template sheets for a range of different assays. As you scroll across the assay sheet, there will be columns and tags associated to the protocols again. So you'll see details for the corresponding data files and metabolite tables. So raw spectral data files are files directly from the analytical platform or instrument, and derived spectral data files include open source converted files or any derived data file you may wish to include. And as before, a primary requirement for study submission to Metabolights is raw data or an open source version, along with the supporting metadata, which needs to be complete, and a Metabolights user account. And of course, you can have multiple assay sheets, for example, positive and negative mode ionization, reverse phase and HILIC chromatography, or LC-GC-MS, 1D proton NMR, or 1D carbon-13 NMR. You can have as many assay sheets, as many assay sheets as you wish. In the 5th tab, metabolites. So this is where you will find information related to the metabolites identified in a study. So each row is a metabolite or metabolite feature, peak, or an unknown. There are columns for structural information, so smiles and inchi, descriptors for metabolites, as well as intensity values and metabolite abundances. These metabolite sheets are also known as metabolite assignment files or MAF sheets. Where metabolites have been identified, the provided compound names are assigned to a specific ID using the KEBI ontology. And this is a controlled ontology, which means that the metabolites you are looking for can be easily identified and linked. And from KEBI, you can get information such as smiles or inchi. Submitters are encouraged to submit further information, so in relation to metabolite identification. So NMR studies will include chemical shift and multiplicity. Mass spec studies will include mass to charge ratio, fragmentation, fragments, modifications, charge and retention time. And for all platforms, it's encouraged that an identification reliability score is provided. And of course, you can have multiple metabolite sheets or MAF files. In the last tab, which is the files, you'll find both the metadata information, which is uploaded information previously described, so encoded in the standardized and open source ISA tab, ISA delimited format. as well as the raw or derived data files. So all public studies and associated data in Metabolights are freely available to download. You can download individual files, or you can have the whole folders. You can search for specific files or file types. And if you go down, there's a section called Audit Files, which acts like a version control to keep track of all changes. While it may not be obvious with the new website and submission tool, Metabolite submissions are powered by, again, mentioned previously, by the ISA format or software suite. So experimental data gets submitted in the ISA tab format. The ISA format, so the ISA format is the Investigation Study Assay Framework, and it really helps to collect and communicate rich and complex experimental metadata. So, your sample characteristics, your technology and measurement types, as well as your sample to data relationships, so that the resulting data and discoveries are reproducible and reusable, aka fair. And the ISA framework is comparable to other standards used in other omics, such as the MAGE tab for transcriptomics or proteomics data. So how to submit a study to Metabolights? You can submit studies to Metabolights through the new guided submission. So that's step by step, and the online editor tool. The online editor tool allows you to come back and edit your study at any stage during the submission process, and this can be found under My Studies page. Working with larger metabolomic service providers or phenome centers, core facilities, as well as companies, we have developed a template-driven submission pipeline for ease of submission of standard protocols and assays. So, for example, we can utilize tools such as MZML to ISA for direct extraction of protocol information from raw data files. All studies are manually curated for high quality. We support submitters through a highly responsive help desk, and the study will remain private between you and the Metabolights team until you tell us otherwise. On the website, there is a quick start guide to help explain the whole process in more depth. So the first step would be to create an account. The second is to create a new study. You submit a study and get a permanent accession, which can be referenced. You then add, edit, and upload data and metadata. You clear validation and you start curation to finally being in review, where you get a temporary private read-only link for peer review, and finally to a publication. We've been working hard to provide a simplified submission process. So as part of this, you can now submit and edit your study completely online without the need to interact with, for example, ISA Creator. And once the guided submission is complete, the study is available in the online editor for further amendments. And you can switch between a guided study view and an editor view using the icons along the sides. You can invite collaborators to be submitters too, so they can update certain aspects of the protocols or add supplementary data files. So what to include in the study? Well, the first section of the presentation, you can see examples of the information found in metabolite studies, and the guided submission will take you through the required information step by step. So it will guide you through the raw and sample data upload, creating assays, and it will provide you with controlled vocabularies or ontologies in the relevant fields. If you don't have all the information to hand, don't worry, you can access and edit your study at any time from your My Studies page, including adding samples, extra assays, data, and much more. For those working in larger labs with LIMS systems, you may be interested to know we're interested in programmatically accessing any part of your study, private or public, from our application programming interface, our API. As mentioned before, once created, a study will appear in your My Studies page. And from here, you can edit using the guided submission or study overview options. Studies can also be downloaded. So the ISO metadata can be downloaded, edited offline locally with Excel. So copy and pasted with Excel and re-uploaded to populate your study. A note on study completion and validation rules. So you must ensure your study passes all the required validations. If a study displays failed, please look at the study validations tab to find out more, right? All errors must be addressed to progress the study, and warnings can help you alert to missing or mismatched information. There are 4 validation tags in the validation tab, which you can be viewed individually using the drop-down menu. So successful completion, information, warning, and error. And as mentioned, you must address all the errors and also address warnings where applicable to your study. Validation rules range from missing data to defined headers, character lengths, unique IDs, file types, referencing, and so on. And the validation box will disappear when all validations are successful. And before progressing your study, please give an approximate date of when you would like the data to be publicly accessible. The curation process requires a minimum of 4 weeks, so you'll not be able to set this earlier than 4 weeks in the future. And once in curation or in the review stage, you can only make changes by contacting us directly at metabolights-help@ebi.ac.uk. So reminder, there are 4 stages: submission, curation, review, and public. And the in-curation step is just a step to assess whether the study is complete and fulfills the requirements of Metabolights. And to progress the study, you'll need to change the study status to let us know it's ready for in-curation. And this can be done once validations are successful. So I just wanted to highlight a few example studies and to navigate from like a user as well as a curator's perspective. And I think this is the best way to learn Metabolights. So first, you will see the familiar reporting of our study ID, the title, submitters, authors, and abstract. So this one is MTBLS881. So metabotype in patient journeys reveals early predeposition to lung injury after cardiac surgery. Actually, I'll skip this one because it's more NMR, and I'll show the LCMS one, the mass spec one. Actually, it's imaging mass spec, but has some LCMS. So again, we will see the reporting of a study ID, title, submitters, authors, and abstract. So this one is MTBLS78. This is SpaceM reveals metabolic states of single cells, and SpaceM is an open source method for in situ single cell metabolomics of cultured cells that integrates microscopy with imaging mass spectrometry. The first thing we'll be able to identify is the organism, as well as the acquisition technology and metabolite data, as well as associated publications. So for example, here we can see it's from humans or Homo sapiens, as well as mice, and it has samples from blank and reference compounds. We can see that the assays are LCMS, as well as imaging mass spectrometry, and we can see that it's all untargeted metabolomics. And we can link out to other repositories, for example, Metaspace, which is where your imaging studies will put their MSMS data. We can take a look at the protocols, so and we can use subheaders to define different assays. We can then look at the files. This is all the metadata as well as the raw files or derived files. and we can download them and we can open them up in Excel. So for example, from the sample sheet, we can see that there are 123 rows. So these are samples, some human, some mouse, some blanks, and these are what we call the characteristic columns. And as we move across, we can see the factor columns. So these are the columns to describe the experimental design. From the assay sheet, again, if we open in Excel, we can see that it's an imaging, a mass spec imaging assay. It has 16 rows, so that's the samples, and they all have identical experimental conditions. And as we move across, we will see each of these details in. And then from the metabolite sheet, we can see that there were 1,446 rows. So these are metabolite features, peaks, unknowns. Actually, the majority here were assigned to a KEBI ID. So then we have the chemical formula, the SMILES, the INCHI, as well as any descriptors associated to the mass spec assay, and potentially any quantitative values. I'm going to skip the reference library and conclude with a couple of slides to say, for any further information about training and guides, please check out the EBI training website. There are now many great reused cases where people have developed softwares that use the Metabolights public studies, which you can use. So for example, the GMPS dashboard, which you'll hear about lots later, Metabolite Analysts with their COVID-19 Metabolomics Explorer, as well as Galaxy and the workflow for Metabolomics group, where you can choose your public study to reanalyze, get the data off the FTP public server, and use it in the Galaxy framework. To browse data, well, public studies from Metabolights are shared or exported with the Metabolomics Exchange. So this is an initiative set up to aggregate and notify service for the Metabolomics community, as well as the Omics Discovery Index. This is a cross-omics aggregator developed through the BD2K. Again, we have our counterpart repository. So there are a number of other repositories for metabolomics deposition. I'm going to add massive to this moving forward. And these guys also disseminate their data through the Metabolomics Exchange. And also Metabolights work closely with these. So this is the Metabolomics Workbench in the US, supported by the NIH. And this is Metabobank in Japan, supported by the DDBJ. An advanced one, what we're looking at recently in Metabolights is the multi-omic study submission. So now we are getting complex study designs, also implementation, for example, big multi-omic studies. So for example, the Elixir implementation studies, Holofood as one example. And we're looking at how you link data, different multi-omics data across the same study. So for example, you would have the same sample ID that would be deposited to biosamples, and then those each information, biological information would then link out for metabolites if you have data for that sample with regards to metabolites. If you have proteomics data, you could then, for that same sample, you would link it to the PRIDE database. If you have transcriptomic data, it would go to ENA, the European Nucleotide Archive. And then if you have transcriptomic data for environmental samples or metagenomics, it goes to ENA, but then also links back into Magnify, for example, if you use the Magnify platform for data analysis. And there are many other resources that Metabolights links to, for example, we said earlier, Metaspace, Bioimage Archive. And also, just to finish, that there are many other data resources at EMBL EBI, so not just focused on chemical molecules and drug discovery, such as metabolites and KEBI, but from genes and genomes and RNA, so the Ensemble database, the ENA, the Nucleotide Archive, Magnify, to the proteins such as PRIDE or AlphaFold, UniProt, to imaging and cellular structure information, genetic variation and disease, and literature and knowledge management. So please do make use of all those resources to help you. We at the EBI Metabolights team, we're a team of curators and developers, quite a small team. So yeah, please do be patient with us, and we are always welcoming feedback. So user input on existing and new features, any features you would like to include, how best to capture assay and metabolite annotations, and what other kind of data you would like us to capture, as well as interactions with other resources you would like to see. Yeah, so with that, I'm happy to end and take any questions.
Okay, thanks so much, Tom, for this awesome presentation. And it's pretty impressive to see how many resources there are from EMBL EBI. Okay, are there questions in the audience? I may can repeat it then for you. I don't know if you can hear people very well.
If you could repeat, that would be great. Yeah. Thanks.
Okay. Right. Okay, seemed to be pretty much clear to everybody.
Cool. Thanks.
So,
yeah.
Okay, let me check the chat. All right, then, yeah, let's thank Tom again for zooming in from the UK.